the centipedes that I have worked with. And we'll start off talking about some of the venom complexity within this group, Scolopendra. And then we'll get into talking about venom resistance using Drosophila later on. And so our lab is, as Darren kind of pointed out, we're interested in some of these evolutionary concepts and how things are changing over time. Particularly, we are interested in molecular evolution. So we're looking at how changes have occurred on the level of the genome or the transcriptome at that nucleotide and amino acid level. And these types of analysis have been done for quite a while in a variety of different organisms, where we can actually take a phenotype that is being expressed and find out the underlying genetic mechanisms and how those mechanisms are changing. So one example of this is in a quantitative trait loci analysis that was done in mice, where we can see here that there are two distinct phenotypes that we see in this one species of mouse, where the mice that live on the beach have this wider coat color here, and the mainland species have this darker coat color. And through this quantitative trait loci analysis, they're able to create these hybrids and then look at the areas of the genome of where these where the coat color genes are being affected. And so through doing these types of experiments, we're actually able to look at the molecular changes that are happening to give these broader scale phenotypes. And so broadly, this is what our lab is interested in looking at. However, it's not always as clear cut of a phenotype as a coat color, where we have a white mouse and a dark mouse. So here I'm showing 3 different fruit flies that are all showing a different phenotypic response to heavy metal toxicity. And so these types of phenotypes are a lot harder to be able to dissect apart and to understand the molecular evolution and the mechanisms that are going on underneath it. However, we have a lot of different genetic tools like that quantitative trait loci, as well as other different genetic experiments that we can do to analyze these complex traits. So instead of looking at just a few specific alleles that are helping to change coat color, we often see diagrams like this, where we can see it's not just one gene affecting the phenotype, but it is a variety of different genes that are interconnected in a molecular pathway that help us to understand some of these more complex phenotypes, like heavy metal toxicity or the resistance to heavy metal toxicity in the fruit flies, like this example here. So that just wasn't quite enough. And so I wanted to actually dive a little bit deeper into it, because I'm more interested in some of the co-evolutionary relationships that happen within nature. And so when we think of co-evolutionary relationships, we're not just looking at that one phenotype, but we are looking at 2 phenotypes for 2 different organisms. So if we look here on the left-hand side, we have Darwin's moth, which has this long proboscis, which is able to go and get the nectar from this flower. And so if we wanted to study this co-evolutionary trait, we would not only have to look at the genetics and the underlying mechanisms for this long proboscis, but also for the flower, and why and how these relationships have changed over time to where these 2 animals are so interconnected. The other example that kind of shows off the complex nature of some of these co-evolutionary relationships is this example on the right, where we have a species of trematode, or a parasitic worm, that actually is goes through a complex life cycle where it is able to infect snails. And so once the snail eats these eggs, the adults will then mature and move into their eye stalks that make their eye stalks look a lot like caterpillars. And it'll actually cause these snails to move to the top of grass stalks, where they're able to be more easily predated on by birds. And so this co-evolutionary relationship goes through a complex life cycle, not only with one organism, which is the trematode, but it also goes through birds and snails. And this complex life cycle has a lot of co-evolutionary relationships and brings about a lot of interesting questions that are not necessarily the easiest to answer, but help us to understand the world around us a lot better. And so one example that I wanted to talk about these co-evolutionary relationships, and one that has been able to, we've been able to learn a little bit more about as far as the molecular mechanisms that are involved, is with the garter snakes and newts. And so here we can see on this left-hand side that we have a garter snake eating a newt. And in this case, it looks like the newt is probably going to be the loser in this situation. However, oftentimes it's the other way around, where the snake is oftentimes going to be the loser of a situation like this. And this is due to the newts having a specific toxin on their skin, which is called tetrodotoxin. And this tetrodotoxin will go and bind some of the outside channels in our nerve cells and make it so those muscles won't fire anymore, and your body cannot send or receive signals. And so it shuts down the entire nervous response that happens. And so these are a very, very toxic newt that can kill not only the snakes, but have known to even kill humans with this type of toxin. And so a lot of work has gone into studying the co-evolutionary relationship between both the newts and the garter snakes. So here on the top panel, we can see that across the western coast of North America, up into Canada, into California, and down into Mexico, we have these spots where we have both toxic newts, which is represented by these warmer colors here, is they have a higher toxicity, or they produce more of that tetrodotoxin. And then on the right-hand side, we have the same map, but this time it's showing the level of toxin resistance in our garter snakes. And so where we see more of this toxicity in newts, we see a higher toxic resistance in the garter snakes, as they are kind of in this relationship and this kind of co-evolutionary war of who can eat who and defend against who. And so this is a really interesting co-evolutionary relationship that, again, through some of the molecular techniques that we have available to us, we've actually been able to understand the exact molecular changes that need to happen on the receptors that make it so the garter snake is resistant to that tetrodotoxin. So here on the bottom, we have the three-dimensional structure of some of these proteins and the exact amino acid changes that need to happen to produce this tetrodotoxin resistance. And so this is a really cool example that is able to show how these co-evolutionary relationships can still be determined at kind of their underlying molecular mechanisms, and how the molecular evolution is changing these two species at not only this molecular level, but also at the more regional scale and across different populations. And so in our lab, we want to do this with venom. And so venom is a complex mixture of different types of proteins, like Darren was describing in the beginning, where in a venom, there can be anywhere up to hundreds of different types of components. However, some of these components are overlapping, but they have some similar protein families that are found in them. So this is from this venom composition that I'm showing here is from the centipede Scolopocryptops sexspinosus, which is this red centipede here, where centipedes have these distinct forcipules, or these little mouth parts at the beginning of their head, where they're able to inject venom into a potential predator or prey. So I was able to characterize this venom when I first got here to Florida State, and we were able to find all of these different toxin families that are involved. And so, like Darren said, these toxin families don't necessarily work in unison, where one does one job and one does another job, but they work synergistically. And so if we look at just two examples here, if we look at this big category here on the top, these are called the adenomycin-like metalloproteases, that their main function in the body is to destroy cells and to kind of open up and cause a big wound in the potential prey or predator. And then these beta pore forming toxins are a toxin that is commonly found in not just bacteria, but in some animal venoms as well. And its main purpose is to kind of poke holes in cell membranes, and that could kind of amplify the effect of these adenomycin-like proteins. So it's possible that these two different types of proteins can work synergistically together. And so with looking into venom, I also want to look at that co-evolutionary relationship, which is venom resistance. And venom resistance up until this point has mainly been looked at in cases of different types of snakes and their predators or prey, or in different types of mammalian predators with their invertebrate prey. And so these studies typically look at kind of a one singular venom component versus a singular mechanism for venom resistance. So again, we can kind of see these different protein families listed here on the bottom, where we see one type of protein, which this red one here is called snake venom metalloproteases. And there has been work that has characterized a specific inhibitor that will bind to these and make it so they're not effective. But these snake venom metalloproteases probably work in conjunction with these lectin-like proteins that are shown here in dark blue, where these lectin proteins actually work with the blood cascade, and the snake venom metalloproteases destroy those capillaries and cause this massive bleeding effect. Where the C-type lectins that are shown here are involved with that coagulation cascade and make it so your body cannot coagulate that blood, and so you end up bleeding out. And so these synergistic mechanisms have yet to kind of been looked at in venom resistance, where we've just kind of focused on that singular mechanism and singular venom component. However, we kind of need that broader complex response where this venom resistance is not just a singular mechanism that's able to provide resistance for the whole venom, but there are likely multiple mechanisms and multiple underlying genes associated with this venom resistance. And so this is where I'm hoping to kind of provide some insight into what exactly is going on with this co-evolutionary relationship. And so just to highlight a few examples of species that have demonstrated venom resistance and how this is a common evolutionary adaptation across the tree of life, we have this example here on the top left, where this mouse is actually able to eat toxic scorpions. So the scorpions that we have in Arizona that are the most deadly in North America, these mice are able to eat them readily. And so there's a singular toxin change that we've discovered that has led them to be able to eat these scorpions. And then if we look at this other pair of species over here, this is a honey badger with I believe a species of cobra, which honey badgers are highly resistant to a lot of different types of snake venoms. And this is what led to one of my favorite papers, which is why the honey badger don't care. And so these animals are resistant to a type of acetylcholine receptor, which is one of the main receptors that is targeted by venom components. However, again, like I was saying on the previous slide, these two different mechanisms here have kind of formed from a singular venom component being investigated to a singular mechanism of venom resistance. And we've yet to see kind of this broad application looking at venom resistance from a whole complete venom and a whole complete suite of venom resistance genes. And so for my project, I wanted to look at kind of the two different components of this. So in order to understand venom resistance, we first have to understand the venom. And so the first part of my project is looking at the molecular characterization of centipede venoms within the order Scolopendra morpha. And so this will kind of be the first part of my talk here, where we can look at the venom and Venom-Gland characterizations that I've done, and then look at the molecular evolution of some of those components that I've been able to identify. The next part of my talk will be focusing in on that co-evolutionary relationship, where I'm going to be looking at mechanisms of venom resistance in Drosophila or fruit flies. So I'll be able to kind of expand on that later, where we'll be using a similar type of analysis like I had in my introduction, except this is called an extreme quantitative trait loci mapping experiment. But this will kind of help us to get down to how these animals are evolving this venom resistance. And then lastly, I'll talk about some future work that I plan on doing and that I'm currently working on, which is validating some of the venom resistance genes that I've identified in AIM2. And so first off, I will start talking about toxin diversity and looking at the venom of these different species within the order Scolopendra morpha. And so for this project, I've been able to do field work across the United States, where we've collected animals down in the Keys here in Florida. We have a few local species that we've collected right around here in Tallahassee, but then we've also collected centipedes from across the southern US, including in Texas, Arizona, and California. And so we have a variety of different centipedes that we've been able to collect and work with. And what happens is we'll go out and collect these centipedes and bring them back into the lab, where then I'm able to extract venom. And so how I'm able to extract the venom is they have those forcipules, or those biting mouth parts, on the front of the organism, where I'm able to have them bite around a metal spatula. And then that metal spatula, since the centipedes are anesthetized and are biting around that metal spatula, we're able to give them a slight electrical shock, which will make their muscles contract right around the venom glands and expel the venom out onto the metal spatula, where we can collect it and work with it. So we're able to do this for a variety of different species, including some very small individuals. So this thumb here isn't a giant's thumb, that's my thumb here. So this centipede is actually pretty small. And it's one of the centipedes that we have locally here in Tallahassee. But once that venom has been kind of expelled out onto this spatula, like we can see here on this right side, we're able to collect that and then save it for later proteomic analysis, or for some functional verification, like I'm doing in the second part of my talk. So then after we get venom, 4 days later, when we're hoping that transcription is going to be the highest, we dissect out the venom glands and isolate the mRNA, and then are able to compare not only the proteomic, or that phenotype of the venom, but also relate that back to the genetics using the transcriptome. And so through this, we've developed a pipeline that we've been able to, I wrote some scripts to automate this pipeline, where we have our centipede here. We're able to get the proteins from the venom directly. We're able to analyze those proteins through mass spectrometry to get their amino acid sequences. We're also able to get the mRNA sequences from their venom glands and sequence those, and then look at sequences that are homologous to other centipede proteins, as well as look at which proteins we identified from the mass spec to be able to label all of our toxin proteins. And so this bioinformatic pipeline I've been able to do with the 26 individuals that I collected on that first map figure. And so this is, we've been able to collect the proteomic data and the transcriptomic data. However, with the transcriptomic data, we don't only get the toxin sequences, but we get non-toxin sequences. And so the first thing that I wanted to do was to see if these species, or how closely these species are related. So I was able to isolate around 700 different non-toxin sequences that were collected from the BUSCO program from the transcriptome. And I was able to generate this phylogeny with those sequences. And so I included mainly centipedes within the genus Scolopendra. However, I included 2 outgroup species. One of the outgroup species was that first centipede that I characterized when I got here at Florida State, which is Scholopocryptops sexspinosus. And we can see that it falls out nicely here on the bottom. The other species that I included as an outgroup is Hemiscolopendra marginata. And so this centipede, we can see clustered within the genus Scolopendra. And so this is kind of highlighting one of the things that needs to be done with centipede taxonomy is that a lot of the phylogenetics that have happened within this group are based purely on morphology. But there's a lot of other genetic mechanisms and kind of cryptic species that could be found in these groups, which we can kind of talk about with some of these other findings that I found within this phylogeny, including potentially looking at the genus classification for some of these organisms. And so most of these were found to be just species pairs. However, there were a few different species that I thought potentially could be broken up into multiple species, and with some more work could be elevated to their own species. So the first one I wanted to look at was the species called Scolopendra polymorpha. And so with a name like polymorpha, it's not surprising that we found quite a few different groups here. So looking within all of our polymorpha specimens that we collected, we have this group of 3 specimens here, this group of 2 specimens that kind of cluster out, and then this group of 7 Scolopendra polymorpha that cluster out down here. And so we have these 3 different groups that through this genetic analysis are probably should be elevated to a new species. However, like I said, we kind of need a little bit more work to be able to distinguish those. And so for my future work, I kind of separated them out and characterized their toxins based on their lineages that we see here. The other species that I wanted to highlight is Scolopendra heros, which again has a very broad range like Scolopendra polymorpha, where we see a mainly Texas clade here, and then we see kind of our Arizona clade here on the bottom. And so this species, again, could probably be separated into multiple species, but we need more sampling and more genetic mechanisms to be able to kind of look at the phylogenetics for these different groups. So these were kind of some interesting preliminary results, like I said. However, I was able to apply these groupings to how I identified the toxins in that pipeline that I just showed you. So going through that pipeline, I was able to identify some putative toxin families. And so I identified all of these different families here, like the beta pore-forming toxins that I talked about earlier in my talk, as well as the metalloproteases that I talked about, as well as some other toxin families that are commonly found in centipedes. And I was able to do this using both the transcriptomic data and the proteomic data. And so I was able to generate these figures with all of that data, where we can look at the toxin family composition among all of these different groups. And so there's a lot of information shown here in this chart, where we have those different groupings of species that I have down here. And then in our colored bar graphs here, we have the different toxin families and their percent composition. And then whether they are male and female across the top. And so, like I said, this is a lot of information that I've kind of thrown out and are having you guys look at, but I wanted to highlight a few of the different groups that maybe look a little different here. So the first group I wanted to highlight is this group right here, which is Scolopocryptops sexspinosus. And so again, this was the outgroup that I used, but it is still within that order Scolopendra morpha. And we can see here that this venom has these distinct yellow bars that we don't see anywhere else on this figure. And so these yellow bars are the metalloproteases that I was able to write about for this species. So we were able to publish a paper on these results where we identified an atomylysin-like metalloprotease that is very similar to snake venom metalloproteases. And this is a unique venom component that is only found in snake venoms, and so far hasn't been described in any other animal venoms besides this one centipede. And so we were able to publish this paper from the work that I was doing when I first got here at Florida State, where we were able to look at the toxin structure and kind of compare it to those snake venoms, as well as describe this new venom component in centipedes. And so this was a good kind of project, kind of getting me into this system here. But then as we're kind of looking across this group, there's one other group that I wanted to highlight, which is this one here, where we actually have a male and female pair that show pretty dramatic differences in their venom components. And so we did have only these 2 individuals that we have the transcriptome and proteome for, but we can see that one group has this orange bar here where the other group doesn't. And this other one is mostly described by venom proteins or uncharacterized proteins that we're not really sure what those proteins are. So for this species, we were able to collect a variety of different individuals from the same location, both male and female. We were able to test their venom using HPLC, or high performance liquid chromatography. And with testing multiple individuals, not just these focal ones, we were able to see that male venom and female venoms are distinct. And so looking at the HPLC and looking at the different peaks that came off of there, we were able to run a principal component analysis that clearly separated our male venoms from our female venoms. And so we were able to describe this sex-based difference in venom composition in centipedes, which hasn't been described before and hasn't been noted in any other species besides Hemiscolopendra marginata. So this was another pretty cool example of just looking at the toxin diversity of some of the results that we were able to get from this work. And so again, I was also able to get, this was looking at the transcriptomic abundances here, but also looking at the proteomic abundances, we see that they're pretty similar to those transcriptomic abundances. And we can see that across the middle here, we kind of have all of the same venoms, where we mainly have these 3 different centipedes here that have venoms that are slightly different than those in the middle here. And there was one venom family that I wanted to focus on a little bit more, which are these cap proteins shown here in teal. And so these cap proteins stand for cysteine-rich secretory protein antigen 5 and pathogenesis related proteins, which is why we just call them caps instead of having to say that every time. But these are recruited into a variety of different animal venoms and have diverse functional roles. These are also highly expressed in other centipede species outside of our lab, the ones that we've studied, but in other centipedes that other labs have looked at. So I wanted to look at how this protein family has been evolving on a molecular level. So the first thing that I wanted to do for this analysis was to look at a gene tree. To see if we could see if there are a lot of differences with all of the cap proteins that I identified within these 26 individuals. So I generated this cap gene tree here, where we kind of see some distinct groupings, where we have kind of this group here on the bottom, this group right here, and then these 2 groups here on the top. And these actually nicely cluster out with what is currently known about these venom proteins from other work, where they've been kind of separated based on their recruitment into venoms, based on their genetic history, as well as their function. And so we have our CAP3 proteins that we only found one of here on the top. And that was what only found in that Hemiscolopendra marginata species, which is that one outgroup species. We have our CAP1 proteins that were found in our other outgroup, which is Scolopocryptops sexspinosus. And these are kind of a more basal cap that they thought were recruited earlier on in centipede evolution. And then lastly, we have CAP2 proteins that I've separated into 2 different groups here. And our CAP2 proteins are widely abundant within the order Scolopendra. And we have 2 different functions with these different cap proteins. So these cap proteins here on the top that are shown here in green are the cap proteins that are associated with serine protease inhibitors. So they actually inhibit different type of proteases, where these cap proteins here in purple are actually calcium channel inhibitors, that they inhibit calcium channel function. And so they're more of a neurotoxic component of venoms. And so these are the 2, it'd be hard to kind of look at the molecular changes within this whole group since they are so divergent. So I just wanted to look at the CAP2-2 proteins, or those calcium channel inhibitors. So I generated a new gene tree here, where this new gene tree has all of these different CAP2 proteins. And we see that we have kind of a nice tight group phylogeny here that still has a lot of different changes. And so I wanted to see if these proteins were evolving under positive selection. Positive selection is often thought of as how proteins in venoms have evolved, where they kind of have these increase in copy number within the genome. And then we see these changes that help to diversify these different proteins. And so I wanted to see if there were any of these positive selection events occurring on this gene tree. So I was able to run an analysis through HiFi, looking at positive selection across the entirety of this gene tree. And with that analysis, I was able to determine that there was positive selection occurring on this gene tree. And so then I started doing some additional tests in HiFi to look at kind of a finer scale. So the first test I looked at was to look at if any of these branches here are evolving under positive selection more than other branches within the tree. And so when I ran that test, I was able to see here that there are a few different branches that were shown to have positive selection that were under more positive selection than the other branches. And those are these two branches here. I then wanted to look at a slightly finer scale again. So I wanted to look and see if there were proteins that were evolving on a site-by-site basis or on the amino acid level basis. And so I was able to do a selection analysis where I looked for evidence of positive selection across the entire protein, which is shown here on the x-axis. And then I have the p-value plotted on the y-axis. And so I was able to identify 5 different sites here that were evolving under positive selection across this entire protein. And so these 5 different sites you can see occur over about a third of the protein. And this was kind of some exciting results because we are detecting this evidence of positive selection like we would have expected in this protein group. Again, I wanted to take it another step and look at where these sites were on the actual 3-dimensional structure of the protein. So there is a new program that came out within this last year called AlphaFold that is able to predict the protein structure based on structures that have already been solved through X-ray crystallography. And so I was able to run these proteins through this program to be able to look at the 3-dimensional structure that these proteins have. And so in this picture here, we're able to see the different molecular structures that are in the protein, where the pink is showing us are the alpha helices of the protein. The yellows are showing those beta sheets, and then the whites are showing the random loops throughout this protein. And so with this predicted structure, I wanted to see where on this predicted structure were the changes happening. Because like I said, they were over a third of the protein. So I wanted to know if they were all occurring on one edge or kind of throughout the entire protein. And when I mapped those selection results onto the 3-dimensional model, we can see that all of those results that are now highlighted here in blue occurred on the one end of the protein and kind of in this one section. And so we are pretty sure that this protein functions as a calcium channel inhibitor. However, we kind of need some more work done with this particular protein to determine the actual function of it and to determine where the binding sites are. But this is probably a high likelihood that this could be either a binding site or a place where other molecules can bind to and inactivate this protein. And so this was kind of a really cool result that we were able to not only see the molecular evolution and the toxin diversity kind of from a species to species, but also within a singular protein family, we were able to track these small molecular changes over time that have happened. And so this is some exciting results that hopefully I can look into further to not only look at other protein families and look at how they're evolving on kind of this 3-dimensional model structures, but also learning how these different 3-dimensional models are actually functioning to see if these changes are happening on binding sites or active sites of these different molecules. And so that's kind of the first part of my talk here, where I've talked about the molecular evolution of Scolopendra morph venom, where I characterized the venom and venom glands from those 26 individuals. And I was able to identify that protein family that was evolving under positive selection. So now we're going to switch gears a little bit and look at more of those mechanisms of venom resistance. So looking at those mechanisms of venom resistance, I was using venom from 2 of the centipedes that I've talked about today, and using those venoms in Drosophila to be able to determine these mechanisms of venom resistance. And so to determine the mechanisms of venom resistance, the experimental design that I used is called an extreme quantitative trait loci mapping experiment, where I took about 600 individuals of fruit flies, which we can see here in the top right, and I injected each of them with a dose that's supposed to kill about 80% of those that are injected with it. So a lethal dose 80. So we take a group of flies, we inject them with this lethal dose 80, and we have about 20% of those flies. So these flies that survived that venom injection, I would then sequence, which turns out to be around 100 flies. I then sequence flies from the base population that were not injected, so as a control. And then after sequencing those, I'm able to compare the allele frequency changes that occurred between the base population and these venom-resistant flies to see which genes are potentially helping with this venom resistance. And so I wanted to pick two kind of distinct venoms to use for this experiment. And so the venoms that I decided to use was the venoms from Scolopocryptops sexspinosus that has that mainly proteolytic type venom that we have here with this yellow bar here. And then I wanted to pick a rather simple venom that mainly contains neurotoxin components, which is this venom here, Scolopendra viridis. And so those are the two venoms that I ended up using for this extreme QTL analysis. So this led to me injecting a lot of fruit flies. So like I said, each experiment that I did, I injected 600 fruit flies, which took me about 3 hours to do, where I would sit under a microscope for 3 hours and inject fruit flies one by one, like is shown here. I did each of these experiments I did in duplicate, but I also did the males and the females separately. The males needed much less venom to be able to kill the flies than the females. And it was about double the amount of venom was required to kill the females. And so I was able to do this for those two different venom types. And these are kind of the initial results that we have that I'm still working through. But this was for that mainly neurotoxic venom that I had. And so this graph again has a lot of information. So I'll first have you guys look here at the LD80 values. So like I said, with the male fruit flies, which are shown here in orange and on the bottom half of this graph, I injected a LD50 dose of the venom that was about half as much as I injected the females with. And so that is kind of an interesting observation in and of itself. However, when we look at the areas of the genome that I identified, so if we look at the graph here in the middle, we have the genome positions or the chromosome positions shown here on the x-axis. And on the y-axis, I have the p-values shown after doing a CMH test to determine which allele frequency changes were most significant. And so if we look at these results, I was able to identify 213 significant SNPs, or single nucleotide polymorphisms, across the genome that corresponded with changes between those control and the venom injected replicates in the females. And we can see those here above this dotted line. However, in the male fruit flies, I was only able to identify these 3 SNPs. And so there was quite a big contrast between the males and the females. And so this was the first one that I did. And so I was pretty excited about these results, saying that maybe it was just that the males were not going to be able to gain that resistance as much since they were so sensitive. And so that was kind of an initial thought that we had. However, when we did this same analysis, but with the venom that was mainly that proteolytic type of venom from Scolopocryptops sexspinosus, we can see that, again, the venom that I injected was about half as much for the males as it was for the female fruit flies. But this time we see the opposite pattern, where on the bottom side here, we can see that I identified 380 SNPs in the male fruit fly lines versus the 54 SNPs in the females. And so this kind of it definitely threw me for a loop, and it threw kind of our whole lab as we were discussing this on why we would see kind of these two different changes happening. And so if we look here at kind of a more comparative view with the two graphs stacked on top of each other, we were kind of trying to think of potential, I guess, mechanisms or ways that this could have happened. And so when we're thinking about venom resistance, there could be kind of these generalized venom resistance mechanisms that could be related, for instance, to the immune response, that there's a foreign protein in the body and the immune system will go and kind of try and get rid of that protein and respond to it. And so those could be some of these common areas that we see between the two groups. For instance, this peak here kind of corresponds to this peak down here, as well as we see this peak here on the end kind of corresponds to this peak up here. But we'll obviously see a lot of changes that don't really correspond on a kind of one-to-one. So these might be more unique kind of signatures that are pointing at the unique mechanisms that are providing specific resistance, not this generalized resistance to venom. And so we kind of have that comparing these two, but then when we look at the difference between the males and the females, where we identified less significant genetic loci up here for the males, and then we identified more for the males in this other venom type, that kind of still leaves us a little puzzled, and we're still trying to work through that. However, one of the genes that we've identified that these SNPs are located in is actually called double sex, which is responsible for the sex changes that we see inside Drosophila that corresponds to some of these different types of changes. And so I'm still actively kind of looking into what is causing some of these trends, but we're still kind of at a loss on some of this some of this data that we've collected so far. And so, like I said, we've looked into a few of the different genes where these SNPs have underlined. We have that double sex protein down here, and we can see that there's kind of these broad genetic mechanisms like we saw in that figure in the beginning, where a lot of these proteins are connected. So all of the proteins shown here in yellow are proteins that I identified within the samples that I analyzed. And then the white proteins are ones that are kind of their first degree interactions. And so we can see that there might be multiple genes kind of interacting under a singular mechanism that was selected for. And so my future work is kind of looking at identifying some of these molecular mechanisms and maybe some of these pathways that might be involved with providing this venom resistance, as well as I want to go and order genetic lines of fruit flies that have these genes knocked down so they're not expressed anymore, and then injecting them and a control line with venom to see if these genes actually do provide some level of venom resistance. And so kind of a validation study with these future lines. And so, like I said, some of my future work, I'd like to look at verifying some of these venom resistance genes, as well as continuing the molecular evolution work that I talked about in the first half of my experiment. And so this is going to require me to inject a lot more fruit flies, which I'm hesitantly excited about. So I'll be under the microscope for quite a bit more time, injecting fruit flies one by one. And that's kind of some of the future work that I had planned, as well as this next year, I actually plan on completing a Fulbright studying some of the centipedes down in Brazil, where I'll be working on some island and mainland populations in Brazil and looking at not only their toxin diversity, but their phylogenetic history, as well as their molecular evolution that is occurring between some of these island and mainland populations. So with that, I'd like to thank the funding sources that we've had through NSF for the grants that Darren has received, as well as through Florida State for some of the grants that I've received personally, as well as for the planning grants. I'd also like to thank my great lab mates and for all the help that they give me, kind of helping me work through some of these analysis and get these ideas kind of coming to pass and being able to talk about them. But with that, I'd be happy to take any questions.
Well, that was great. I had a question. I'm not sure it. I think you probably said it and I just missed it. But the male flies had like an LD80 of 50% for both of the toxins you tried, and you knew that going into the experiment. Why? I guess I didn't understand why that was. Why'd you start out with half the dose?
Yeah, so starting out preparing for these experiments, I had to determine a lethal dose 80 to use. And so when I was determining this lethal dose 80 to use for the fruit flies, that was this variable population, we noticed that males needed a different dose than females to get that 80%. And so that was kind of some preliminary results that I needed before even conducting the experiment.
So Schyler, looking at this same graph here, you don't have that many dots. I mean, typically, you know, the bottom of all these graphs would be just chock full of SNPs. So what's determining what makes it as a dot here as opposed to just not showing up, or are these all the SNPs that were tested?
Yeah, so there is, you can kind of see these two lines that go right on the 0 axis here. So with the CMH test, I tested, I don't remember how many, I think it was in the millions of SNPs. And then through the CMH test, I kind of narrowed those down, but then I did a bone Ferroni correction that really kind of pushed that noise on the bottom down to that 0 line. And I'm just looking at those highly significant SNPs here.
Okay, so there were tons of tests and you just not really having them show up.
Yep, they're just right there at that baseline as kind of noise. And so with testing for correcting for that multiple tests, with the bone Ferroni kind of made those go down to 0.
So I have another more general question about the complexity in these venoms. I mean, it's true of snakes as well as centipedes. I mean, there are hundreds of proteins in a lot of these, and there's generally held to be a complexity barrier to adaptation where you really can't optimize too many things at once. And I'm wondering if you or anybody else has tried to fit models that distinguish how many of these or how many dimensions of these sets of proteins actually seem to be being influenced by selection as opposed to just kind of a lot of them just along for the ride or drifting or whatever. Is there any work that you know of that gets at that question?
Yeah, there was a paper that a collaborative paper that our lab came out with that was actually looking at venom complexity and in these venom complexity within these different types of toxin families, and relating that back to prey diversity in rattlesnakes. And so there has been some studies looking at how diversity of toxin components relates to some ecological effects. But I also believe there has been some work done looking at kind of those signatures of positive selections across these different protein families, which is why there's one kind of paper that stands out that has talked about lineages that are older, not having as many signatures of positive selection, where some of the newer lineages of venomous animals have this kind of signatures of positive selection. And so it's kind of a trade-off, but there has been quite a bit of work on this kind of complexity of venom.
Cool.
Schyler?
Yeah.
Nice talk. Were there any significant SNPs that were the same between males and females? It's quite remarkable that it seems like they're completely unrelated.
Yeah, so there were some SNPs that were the exact same SNPs identified in males and females, as well as there were even more SNPs that were found to be within the same gene. So not the same SNP, but within the same gene as males and females. But there is still quite a bit of difference between the males.
So do you think it's worth focusing on, right? Do you think it's worth focusing on those proteins that
seem to be selected in both males and females as being more promising?
Yeah, so for my validation studies that I plan on doing for the last part of my dissertation, I plan on looking at some of those SNPs and some of those genes that are common between males and females, but also common between the two different venom types, as well as some of those components that are distinct. So kind of hitting at both sides to see kind of which ones might have a more insightful impact on venom resistance.
So Schyler, you probably talked about this and I missed it, but what are the natural prey and how much do they vary amongst your different species?
Yeah, so the natural prey varies quite a bit between these different species. So let me come back to this slide here. If we're looking at this slide, obviously the prey found in the Keys is going to be very different than that found in the middle of Texas, where there's also size differences. So these centipedes that are found in Texas have both vertebrate and invertebrate prey. They've been found to eat lizards and other various types of invertebrates, but most centipedes are known to be generalistic predators. We don't know a lot about their specific diets, but that's actually one thing that we're writing a grant to kind of look more into right now.
Going to do eDNA of gut data or something?
Yeah, that's kind of the idea that we're going to try and do on these, because no one has kind of done that work on centipedes yet.
Do you think that could help explain some of your venom variation patterns that you have?
Yeah, so I think that could relate back to kind of the toxin diversity that we have. And again, going back to David's question, venom complexity and tying that venom complexity back to diet complexity. And I think that could be a very interesting potential correlation to look at.
Cool. Really wonderful stuff.
Any other questions? All right, well, thanks everyone. Thanks for that. Great, as promised, that was a lovely end to our semester. So thank you.
All right.
All right.
Don't forget to stop the
Well,
recording.
I will stop the recording. Can I just ask, I still don't, when did you expect males and females to be that different? Like, were you surprised when you got that result, or was that like you did it because you kind of knew that's all the case?
We knew that injecting males and females, that males needed the lower dose. However, kind of these last graphs that I showed here, we did not expect kind of these dramatic differences between the two venom types, where the males are almost nothing in one and then the females are almost nothing in the other. That was a complete surprise.
Okay.
Yeah, it wasn't surprising to get the sex difference. It's surprising to get the sex difference in opposite directions.
Right, for the different kinds of
Yeah.
toxins. Yeah, super cool. Okay, all right. Well, I'll send you, I'll stop the recording, but I will send you the link.